Thank you very much, first for the introduction, secondly for the invitation, and also James and Nick for inviting me to the oldest society of Cambridge, which is really a great honor. So, I have done my career since 1981 on DNA damage and repair, and when reaching retirement, the results were so promising that at retirement age, I started 2 other laboratories next to the one that I have in Rotterdam. So I am affiliated with the Erasmus Medical Center already for a very long time, but also started a laboratory in the Princess Maxima Center in Utrecht. In Utrecht, children with cancer, I'll come back to that later. Also a new lab in Cologne. In Cologne, and I'm also part of the Oncode Institute, but I have no commercial connection, so I'm completely free to talk about the topic, DNA damage, cancer aging, and as you will see in my presentation, the unexpected impact that nutrition can have on medicine. So this is a picture of Peter Bruegel, the elderly, in medieval times. Now, in medieval times, people didn't get old. Actually, as you will see on this detail of the picture, you could have an infection that wandered through Europe, and within weeks, 1/3 of the people had died. So, in medieval times, people didn't get old. However, since 1840, the life expectancy of the country with the longest one in the world, and now for already several decades, Japan, a linear increase. Every week, a weekend is added. If you are born 10 years later, your life expectancy is 2.5 years longer. We have, although now, evidence that it will level off at 120. However, I predict that when we apply what I will show you later, that we might reach 150. And I can show you the evidence that we will have about that. But that is something that will be apparent only in 200 years. So it's now safe to... But aging, of course, is not all of a blessing. There are numerous aging-related diseases, multimorbidity, as you all know, cardiovascular, diabetes, Alzheimer, and so on. But if you turn in terms of money, hair graying and hair loss is even more important, and the most money is wasted on the wrinkles. But still, all of those features are linked with aging. Wow, that is important, but very paradoxically, when I started in 1981 working on DNA repair in Rotterdam, we didn't work at all on diseases that are very common, that all of us, when we reach the age we will have. In old people, we started precisely the opposite. We started with very rare diseases occurring about 1 in a million, or some of them more and some of them less, with children. They are all due to defects. So we started at the opposite spectrum, and I'll show you later how we ended up in the other direction. So one of those diseases is this child. I know the parents of Natalie. She has a disease which has an awful name, a very rare, 1 in 100,000, trichothiodystrophy. And it is very remarkable, and you can recognize it immediately. These children have brittle hair, brittle nails, scaly skin. That is not a problem. You can become 80 with that. But Natalie became only 2.5. And she has brittle hair, but as you may be able to see on the skull, but not that brittle. But what happens, these children get also pneumonia often, and in one night, completely bald. All hair on the cushion. And when she recovered, she got back her brittle hair, but not that brittle, and then it happened again. Very weird. I'll show you what we know and what we learned from these diseases and how we can explain this completely. So Natalie became only 2.5 and suffering from a DNA repair disorder. And now let me first explain you DNA repair. What is repair and why is it important? So DNA that keeps the secret of life, all the instructions for all the proteins are in the DNA, very precious molecule, only 2 copies in the whole cell, is constantly damaged from outside. UV, and already Nick presented a lot of consequences of all of these treatments, chemical agents, chemotherapy, for instance, cause interstrand crosslinks, radiation causes breaks, UV causes these types of damages, but there is another very important source that's constantly there. Of course, the exogenous sources you can try to eliminate or reduce, but reactive oxygen species, alkylations, aldehydes, alcohol contains also, mechanical stress, changes in the cell shape because the muscles, the heart, the vessels, constantly, if you change the cell shape, the nucleus is very delicate. So the nucleus will also deform. And that causes DNA damage. And water reacts with DNA spontaneously. Every cell, every day, you have 10,000 abasic sites in every cell, every day. So we think that every cell may experience up to and maybe even over 100,000 lesions every day. And of course, DNA keeps the instructions for life. So if the DNA is damaged, the metabolism will be affected. And there are 2 extremely important consequences in terms of health. One of them is that if your DNA is damaged and you replicate the damaged template, there's a higher chance you make a mistake, so that you make a mutation, or that the break, a single or double strand break, ends up in a chromosomal aberration. And that is the main cause, as already Nick showed in the first presentation, of cancer. It starts almost always with a DNA damage in some cell that unfortunately activates an oncogene, tumor suppressor gene is inactivated. That's one step in the carcinogenic process. So cancer starts in most of the cases with DNA damage. However, there is another effect which we have very much developed and worked on, namely if you have an interstrand crosslink, a bulky adduct, an intrastrand crosslink, they will perturb transcription. If you have a gene, you need to make an RNA copy of the template strand, then, if there is a damage in that template, or if the 2 strands cannot separate, then you're blocked. So there is effects on transcription and replication equally, and that causes transcription stress and replication stress. And I'll come back to transcription stress particularly because that's something that we discovered very recently. So that leads to, if a cell is dividing and has too much damage, it may make a mistake, and then it ends up with cancer and the whole organism will die. So cells have a mechanism to stop transiently, try to fix the damage before you continue. Or if the damage is too severe, permanently arrest. Then that cell will end up in a senescent state. Also, if transcription is affected, transcription is needed for the function. So if the transcription is affected, the function declines. And also cells may die. And those all, we think, are the main cause of aging. And I'll come back to that later. Fortunately, we are all equipped with a very sophisticated network of repair pathways that tries to handle this problem. However, transcription stress, I'll come back to that later, is very important to try to cope with because, it's not always. So there are many different types of damages. Not all can be repaired by one pathway because they are very different in nature. So there are dedicated pathways for subclasses of lesions in the DNA. So nucleotide excision repair is a pathway that deals with distorting, base pair disrupting lesions, whatever, thousands of different configurations. They are recognized. UV can induce them, chemicals can do. That is a pathway that recognizes distortions. Transcription coupled repair, that is there just to rescue transcription, but it is blocked by a damage. And so on, you have base excision repair for subtle lesions. Homologous recombination and end-joining are 2 of the multiple solutions for a double-strand break, which is very difficult for a cell to handle. And interstrand crosslinks are even more difficult, and there are multiple pathways there as well. What I will highlight is the top 2 as an example. That's just for the problem of DNA damage and repair, but not at all precisely linked with those pathways. The other pathways equally are important. How does it work? Well, we have we have made a lot of contributions to understanding this pathway, nucleotide excision repair, which operates genome-wide, global genome NER. It starts by a damage, for instance, induced by UV light that distorts the helical structure, but numerous chemicals do the same. And in our lab, we have found that the XPC protein is the primary damage recognition factor. It's a protein that binds to DNA, but tries to put its finger in between the strands. And if the DNA is normal, it will not get the finger in between and dissociate and bind somewhere else. Unless it really encounters a damage where you can put your finger in between, and then it stays. And then it recruits a complex of proteins that we discovered together with the French lab, Jean-Marc Egli, 10 proteins in one complex, TF2H, which has two helicases, XPB and XPD. XPD comes back later, so keep it in mind. Those two helicases, they can locally open the DNA, and then XPA is another factor that will try to identify and stabilize this intermediate. And then RPA is another single-strand binding complex of proteins that prevents the re-annealing, because DNA wants to re-anneal, so you keep it open. And then the surgeons, XPF, ESCC1 on the 5' side, XPG on the 3' side, they cut out the piece that contained the damage, and then the gap is filled in with replication. So logical steps of events that occur with this pathway. Almost half of all of the genes involved have been cloned or identified in our laboratory in Rotterdam. This is global genome NER, but there is a problem, because some damages are difficult for this system to recognize. They can persist there for days, for weeks. And if such a lesion, also induced by UV, and UV induces multiple lesions, but the main one is very difficult for global repair to identify. So that persists for long. And if it occurs in the transcribed strand of an active gene, the polymerase bumps upon it, cannot continue, then you need transcription-coupled repair. So there are proteins, the CSB protein that's also cloned in our laboratory, identifies a stalled polymerase, recruits other factors, and then it ends up in the same repair reaction. So transcription-coupled repair only for damage that actually blocks transcription. Global repair looks everywhere for damage. All of the genes that are, all of the proteins that are indicated in pink, there are children that are born with a defect in those genes. And these diseases are abbreviated XP, CS, and TTD, trichothyroidystrophy, that already introduced. Besides those, particularly our laboratory has almost made all the mice. Almost all the mice in this pathway were made in our laboratory, and I'll show you some of them. So every gene with an M, there is a mouse running around. So the diseases. XP stands for xeroderma pigmentosum. Bob Johnson has done a very nice work here already a long time ago. XP, xeroderma, a dry, Perkman-like skin, like you see here, actually a skin that is aged, because these patients are very sensitive to sunlight, to UV. UV damages your DNA, and they cannot handle the damage, and as a consequence, all of these diseases actually are highly UV sensitive. But and you see also the pigmentation abnormalities in sun-exposed parts, too much, and also too little pigmentation. So that is the disease, but the most important aspect is they have an over 1,000-fold increase in skin cancer. This is already the beginning, but children, when they are not exquisitely protected, and this is a child from Lebanon, not recognized, already by the age of 7, many cancers that occur. And this is a mild form. There are children 10 times as worse at this age. Thousands of lesions in the skin, in sun-exposed parts. So that is due to a defect in the global genome system that looks everywhere for damage. I'll come back to that later. So another disease, which is very different, is called Cockayne syndrome. Equally very rare, 1 in 100,000 or less. And these children actually no cancer. Not 1,000-fold even, they have less cancer than normal. in the mouse, we have found. But instead, they have very severe neurodevelopmental abnormalities and an average life expectancy of only 12. It's traumatic disease. This is Baptiste, a French patient. You see already from his posture, is not normal, neurologically already compromised. And now look at Baptiste 4 years later, very small, needs glasses, needs hearing aids, cannot walk. These children cannot speak. And Baptiste became only 10. And this is his sister next to him, 1 year older. So it's really a very devastating disease for which we have nothing. But I'll come back to that in the end. Now, this is always due to a defect in, if the transcription-coupled repair system is affected, then you have cocaine. But there is this other disease, which is called trichothiodystrophy, the brittle hair disease. Well, this is such a patient, many features in common with cocaine. Also no cancer. It's not exactly the same, but it is very similar. And this is always due when the two pathways are not completely, but partially affected. The brittle hair has nothing to do with the repair defect. That is due to a gene expression problem that I can explain to you, that we have discovered using mouse models, but that's trivial. The other features, they are, of course, you can become 80 with brittle hair, but these children, many of the TTD children, don't get older than 5 or 7. So you have many different disease outcomes with defects in one repair system. How can it be? Even you can have mutations in the XPD helicase that opens the DNA. Some mutations in some patients cause XP. Other mutations in XPD cause cocaine and XP. Yet others cause TTD. Again, other mutations cause TTD as well as XP. And yet others cause a disease which is called cerebro-oculofaciosskeletal syndrome. Children do not get older than 1.5. So one gene and all these different outcomes. How can that be? Well, one way is to try to make a mouse precisely in the same mutation as these children. XPD is an essential protein. I'll show you the TTD mouse. XPD is an essential protein, so you cannot completely inactivate it. Then there is no transcription, because TF2H is a transcription factor. It's multiple purpose. It's for opening promoters, but also opening DNA in the context of repair. So if you cannot open promoters, there is no transcription, and so that is no life. But point mutants are viable. And some point mutants give TTD, and others give XP, and yet others give XP and CS. So we made precisely the same point mutation as in this patient in the mouse, and this is the mouse. So at position 722 in the XPD helicase, the normal DNA encodes arginine, R, but now if you're born with a mutation in that codon, no tryptophan, unfortunately, W. If you make that same mutation in the mouse, and we were the only one that succeeded in this operation because it's very delicate, you see the mouse has also brittle hair, scaly skin, and brittle nails. So the mouse, at least in that regard, was a good model for the disease. So we wondered, maybe this mouse can help us to understand the other problems, which are much more important, because these children don't get older. None of the children with that mutation became older than 5. So it's in humans extremely devastating. And then there was an eye-opener. I still recall the moment that the PhD student, Jan de Boer, in our lab, who succeeded in making the mouse, came in, he said, the mice become gray. Gray? Are they aging? No clinician, this is a very rare disease, children with puzzling feelings. Nobody had made that conclusion, because of course you cannot investigate everything, but we started to investigate everything in the mouse. So from DNA repair, now we moved into aging. So how does aging look like in a mouse? Wow, a normal mouse in the lab, we have to keep, they keep them very well, can become 2.5, almost 3 years. So your entire PhD is almost gone when you're when your cohort has reached the age. But our mice, they showed kyphosis, cachexia, sarcopenia, and the mouse actually revealed to us that TTD actually is a premature aging syndrome. And in a mouse you can look at any moment, at any organ and tissue, it is segmental. So not all the tissues actually are accelerated in aging. Very paradoxically, some tissues are even delayed in aging, but the majority is accelerated. Cataract in the eyelids in TTD mice, less cancer compared to the control, which are genetically identical, except for this one mutation. So this mutation lowers cancer incidence at the expense of too much aging. And so this is this shows a number of features. So there's a reduced life expectancy, 1.5, which is still for the mouse perfect. Actually relatively milder compared to patients, but there is cachexia, sarcopenia, early infertilities, cutaneous symptoms, skeletal abnormalities, kyphosis, I showed you, osteoporosis, osteosclerosis occur, progressive neurological abnormalities, lymphodepletion, timing involution, liver and kidney, all show features of accelerated aging. But segmental, only the tissues that do not frequently cell renew, that do not frequently divide, the post-mitotic tissues, those are the ones that have accelerated aging in this mouse. And in retrospect, if you look in literature, some doctors had found, oh yeah, that looks like there is osteoporosis in this child. But yeah, these children have so many things. So before you can conclude that it is aging, yeah, you have to investigate much more. But there is no question, TTD children also suffer from accelerated aging. So humans and mice are not the same, but this mouse is actually a very accurate model. We found in the mouse clinical phenotypes that the clinicians had not yet found in the patient. So we looked in the liver and we said, this liver is aged. When we asked clinicians with the patient, can you have a look at the liver? And all of them came back, yes, liver, the kidney, the blood were found in the mouse before it was found in the patient. So TTD is not primarily a developmental disorder that people thought before, it's actually aging that occurs at the moment that the development is not yet finished. So that's compromising the development. So we wondered, now what would happen if we have made many mice, so when we make the repair defect even more severe by crossing the TTD mouse with an XPA mouse, for instance. Now, we did. XPA mice are cancer prone. Particularly if they are with UV, then they get skin cancer and have some other cancers too, but not, they can still become 2.5. They have a little bit of neurological degeneration. But otherwise, the TTD mouse actually here, that's the mouse that lives about 1.5 years and is actually overprotecting from cancer at the expense of too much aging. And the double mutant XPA TTD, that is a mouse has kyphosis already at the age of 3 weeks and lives about 5 to 8 weeks. So the severity of this repair defect is associated, correlates with the acceleration of a segmental aging phenotype. The more severe the repair defect, you go from 1.5 to only 5 weeks. So we published this, and it is not the only mouse. We have made CSA mice, CSB mice. We have crossed them with XPA, with XPC. We have made XPB mice, XPG mice. They all come down with the same phenotype. Always we see the same type of aging, not in all the organs precisely the same, but it is exactly the same. So when we published this in Science in 2004, wow. The aging community, they couldn't swallow this. One mutation and then all of these aging no, that cannot be true. So editor-in-chief, Rich Miller, published in Aging Cell, 2004, a poem, Snippety, Dr. Jan Hoeijmakers, tired of working with slowly aging beasts, lops out 1-deoxyribonucleotide, now his mice age just as quickly as yeasts. Wow. Now, after more than 10 years, own students use our mice for research on aging. So, and the field has now considered DNA damage as an important cause of aging. But we think it's the main cause, and I'll show you the evidence. So now I show you a mouse where aging is all round, because it has 4 repair defects. The previous one had 2. Now we have a mouse with 4 repair defects. And now aging is not segmental, not only the postmitotic tissues, but also the proliferative tissues. So the bone marrow is also, these mice get anemia, but they show very severe neurodegeneration. When the mice are born, they can see, but they become blind. When they are born, they can hear even the high frequencies. When they get older, they lose them and then get deaf. The cognition, I'll show you motor performance later on videos. Hypertension, vascular stiffness, cardiomyopathy, kyphosis, as you can see. Liver, the kidney, the fat tissue, the muscles, the skin. There is urinary incontinence, the stem cells get exhausted. Overall, a frail phenotype. Even the mouse can, this is not a knockout mouse. This is the ESCC1 gene, was the first human repair gene cloned in the world. That was done also in our laboratory in Rotterdam. And we made a hypomorph mouse. So not a complete knockout, because that is very severe, but a hypomorph mouse, that's the delta mouse. And that becomes 4 to 6 months. But has all this kind of features of aging. And even now, there is recently, in recent years, Steve Horvath has developed an aging clock, a methylation change in the course of aging that is reflecting the biology of aging. This is the only, this category of mice is the only category that has an accelerated epigenetic aging clock. All other mice that have a premature aging phenotype don't have this, at least up till now, but this is the only one that has, and also the glycage clock in serum is also accelerated in these mice, and by the way, also in patients. So this is 4 repair defects. These are critical for all organs and tissues, also the ones with high proliferation. And it's typical, the multimorbidity that you see with normal aging. You never age in one organ or tissue, only you age over the whole body. And it shows what spontaneous DNA damage, we didn't explicitly expose these animals to anything, apparently can do if you cannot properly repair. But even if you can repair, then still, sometimes repair is there too late. Sometimes repair makes a mistake. Sometimes there is damage that cannot be repaired. And also with aging, we think every process goes down, repair will too. And then you will see later what are the consequences. So we think this mouse actually is an extremely valuable mouse for neurodegeneration, for Alzheimer, for Parkinson, for which decades of massive research with pharmacy and in academia has not yet yielded anything that works for Alzheimer or Parkinson or Huntington. We think this mouse, because aging is the most important risk factor for Alzheimer, for Parkinson, for Huntington. You never are born demented. That takes time. And aging is important, and that is the one that is accelerated in the brain of these mice, and I'll show you later what parallels there are. But to explain it, so actually this mouse combines at least 3 human genome stability disorders. Cockayne syndrome, for transcription coupled repair, global genome NER for xeroderma pigmentosum. When these are combined, you get a dramatic synergism. You saw it in the double mutant. But also, there's a cross-link repair defect, Fanconi anemia. Fanconi anemia removes cross-links, and cross-links are important for cells that divide, because they have to take the strands apart. So this defect causes also the proliferative tissues to show accelerated aging. And there is also a defect in another pathway, for which we do not know a human disease yet, but I presume it will exist, single-strand annealing of persistent double-strand breaks. So one gene involved in multiple pathways, and now we see aging everywhere, both in the postmitotic as well as in the replicative tissues. What we also found, and that was a very important observation, when we measured the activity of all genes in mice that have accelerated aging compared to genetically identical wild types born at the same moment, the same food, the same air, and compare the rapidly aging mouse to the normal mouse, in the liver, in the kidney. And when we did that, the first thing we saw on the computer, I still recall, was the IGF, somatotrophic axis, growth hormone. This axis is down. All these short-lived repair mouse mutants, they suppress the growth hormone IGF, somatotrophic axis. And that is an axis that is very important for aging. And instead of putting energy in growth, now they prioritize resilience mechanisms, antioxidant defenses, immune system. They are all boosted. So actually, we think this is a response that, at the expense of growth, you invest into resilience, defense, maintenance, to try to live longer. And actually, that explains, of course, why the mice and also these children are very small. They are constantly suppressing their growth to try to boost, the resilience mechanisms. And the mice are also very tiny. I'll show you later. So that explains why they are small, but we call this a survival response. The animals try to survive, to cope with the damage, and to try to at least become adult and deliver progeny. But that response resembled, at the gene expression level, calorie restriction. If you have too little food, the body is very wise. It doesn't waste it to growth. It now invests into resilience because you have to survive the starvation period. So, and actually calorie restriction is the only universal intervention that delays aging. In yeast, in worms, in flies, in rodents, in non-human primates, and no question in humans as well. So actually, indeed, this parallel shows that what the animals are trying to do is what calorie restriction also tries to do, is to suppress growth and to upregulate your defense mechanisms. So then we wondered, we give the mouse food all the time. So what would happen if you would apply calorie restriction to these mice? Yeah, they are tiny. Actually, these children, they get extra food because they don't grow. So, but we wanted to know, what would calorie restriction do in these animals? And then there was an eye-opener. So this is the survival of ad libitum mice, so that had unlimited food. ERCC1 delta. So the first mouse dies at 12 weeks, 4 months, and the last one 6 months, 27 weeks. So that is the normal survival of these animals when they had normal food. Now what we did, at week 7, reduced calorie intake by 10%. Week 8, 20, week 9, from 9 onwards, 30% less calories. And then look what happened. And then this happened. The mice lived 3 times as long from the moment that you put them on a diet, median maximum lifespan, 3 times, 200% lifespan extension. So, wow. But of course, what about health? Because, if it gets even worse, then it's no help. But all features of accelerated aging were dramatically delayed. Liver, kidney, immune system, retina, vascular system. But the one that benefited most, actually what is also the cause of death of the animals, is the neurodegeneration. The neuronal system dramatically improved. When the mice had tremors and we put them on diet, tremors were gone. So I'll show you some movies. So this is, these are 2 little mates, 16 weeks old. This one had food all the time. That one was on diet from week 7 onwards. Neurologically, this one is already so much compromised that it cannot control the muscles. It shakes. It shows these spastic contractions. You see, it cannot control the muscles because the neuromuscular junctions are very poor. So, and it cannot keep balance. So, and these mice die from that. But this mouse, equally old, now thinks, oh, I get food, is very excited, but neurologically in a much better shape. It runs around. It's not an HDAD mouse, by the way. It's normally very quiet, but in neurologically much better shape. So you can put these mice on a rotating rod. So this was the one on the diet. This one is the one that ad libitum that couldn't walk. This is the wild type on diet. And this is the wild type, the fat mouse that had unlimited food, that could eat as much as it wanted. Now I'll start the movie, and then it starts to turn around and go faster and faster in order to see how can the mice control their muscles. And you'll see in the first round already this one drops because, yeah, that couldn't walk even. Now 3 minutes later, the fat one has dropped, and now in this case, the wild type starts to fall down, and this one is still running. Well, the mouse is a little bit smaller, and in this movie, that one stayed longer, but normally no difference if you do it between the mutant and this one. So the difference between this and that one couldn't be bigger, because in the first round, this one drops, and that one stays on till the very end. So calorie restriction works better than medication ever would be expected to achieve in this disease. And instead of, if this applies to children, instead of 12, they could become 36, but also be in a much better condition. But of course, mice are not humans, and these children, yeah, they get extra food. And they are frail, they are fragile, but our mice are frail and fragile too. So when I argued, actually in Manchester, there is an annual meeting from patients, parents, and clinicians, and that these children should have less food. I've argued for 3 years, and I was accused of criminal activities. And parents were warned not to listen to me. But fortunately, in France, another doctor believed our results, and the parents, I had already explained several times, our idea about it. If you fast or if you have less calories, your body is more investing into resilience. That's beneficial. So we started with this patient, Emma. She is a TTD child. You will see she has brittle hair. She is 6 years old. She cannot walk, talk, read, write. 1150 kilocalories directly in the stomach, because these children actually zero appetite. It's very difficult to get a single spoon in such a child because it spits it out. So you will see, this is what Emma barely can do. Emma can barely crawl. Like the mouse, she shakes, you see? Also her neurons have already declined so much that she can barely walk, crawl, never walked, barely crawl. And it is progressive. It will decline, and yeah, many of these children don't get older than 5 or 7. So, and you see also the brittle hair, by the way. So then this happened. So Emma developed tremors. I hope that the movie doesn't stick, but you see she starts to shake. Oh, now the movie starts to shake too, but she's trembling. She's trembling the whole body. She could not put 2 Duplo blocks on top of each other. So severe. And this is a very bad sign, because then you get paresis, first from the legs, then from the short extremities, and then swallowing problems, and yeah, then it's finished. So prognosis is very unfavorable. So we discussed it, and what we decided to do is to reduce calorie intake in steps. You can do it carefully. 1150, go to 1050. If things go well, we can continue to 1050, 1050, and then to 950, and to 850. So, and then we started 28th of May, 2018. And within 12 days, Emma could build the Duplo again. Then, I got this movie. For the first time in her life, Emma started to walk independently, key balance, walking. She could also not talk, by the way. She made sounds, but yeah, what did it mean? Now you will see. Listen. She can talk, she can pronounce words for the first, and make a head over heel. She started to write her name, Emma. It's not perfect, but for somebody that was shaking so much that 2 Duplo blocks on, now she starts to try to write. She doesn't need diapers. She's starting to learn reading. So, she is bicycling. She is enjoying life. And now, already, close 28th of May, this year, 6 years. So, actually, she has doubled her life expectancy and still doing great. So, calorie restriction dramatically improves health and quality of life, extends lifespan of these children, no doubt, and now the rules have been completely reversed all over the world, and all parents report benefits from lower calorie intake. Now, this is the first treatment of any repair syndrome, safe, globally applicable, affordable, because it saves money, and it is applicable to everybody, every child in the world. But what is the mechanism? How can it be that this child is improving so much? Wow. What we found, we noted that the mutants that involve transcription-coupled repair, they show premature aging in post-mitotic tissues, the neurons, the kidney, the liver, which suggested, well, maybe there is something wrong with transcription in these animals. So we developed a method to measure transcription in a living animal, in the liver and in other organs. So what happens to transcription in these mice? And now I'll show you, and I hope that it's visible. What we did is we did EU injection intraperitoneally, and then 5 hours later we sacrificed the mice, and then we look how much EU is incorporated in the liver nuclei. And you can visualize EU, in this case by red stain. So this is a wild type liver from a mouse of 4 weeks, 16 and 20. There is a bright red stain that indicates there is a lot of RNA synthesis in the wild type. That's this. Now, in the mutant, ESCC1, at 4 weeks, you see already large nuclei, which indicates polyploidization. That's an aging phenotype in the liver. But also there is lower levels of RNA synthesis. If you look at the amount of EU, but at 16 weeks, there's even less. And barely visible at 20 weeks. So you get a dramatic total decline of overall RNA synthesis in this mouse mutant at the age, 20 weeks is old for this mouse, so it may live maybe for 3 or 2 or 4 weeks. But then it will die. But there is a dramatic drop in overall transcription of the genome of this mouse. So we wondered, these are the mutants in transcription-coupled repair. Normal mice and humans are not defective in transcription-coupled repair. Otherwise, you would have cocaine or TTD. So we wondered what happens with normal aging in the mouse. And then, now you have to look at the green color, but that doesn't matter. This is the amount of RNA synthesized in a wild-type adult mouse liver. This is a polyploid nuclei of a wild-type 2-year-old mouse. You see polyploidization, but also nuclear invagination. This is aging, but there is also clearly a lower level of RNA synthesis in a normal-aged mouse. So that shows that, and if you can quantify that, there is no question that with aging, also in normal mice, we see that transcription goes down. So that shows the value of these mice. We didn't know this for wild-type aging. We started first in our mouse mutant and then discovered what we found there also happening in normal aging. So these mice are extremely valuable for aging. There is not, a primrose path to aging and, throw it in the garbage. These mice are extremely important for understanding aging. So when it is DNA damage that causes this transcriptional decline, you can make a prediction because damage is stochastic. So if you have a very short gene, the damage levels have to be high to have one damage in a short gene. But if you have a gene that is 20 or 100-fold longer, which we have, then there is much more chance to have a damage in a long gene compared to a short gene. So we wondered, this drop in transcription, is that gene length dependent? So are the long genes the one that goes down more and the small ones not? So we looked for gene length. So we investigated whether long genes are preferentially suppressed in accelerated and also in normal aging, because there we see the same phenomenon. And now I show you an experiment. For the first one, and I'll show you, I have no time to show all of the other details, but what we did is we compared a wild-type liver expression profile from a normal mouse with ad libitum food and compared that wild-type to a wild-type mouse on dietary restriction. With dietary restriction, many genes go up and many genes go down. And we simply looked the genes that go up, what is the size of those genes in the wild-type? And the genes that go down, what is the size of the down genes in the wild-type with dietary restriction? And these are the black lines. So the up genes, most of the genes, the top is between 10 kilobase and 100 kilobase, which is the normal distribution of genes and length in the genome. And the down genes are equally. There is not much difference between up and down genes with dietary restriction. They are equal in length. Now we looked in ESCC1, the mouse that showed accelerated aging, ad libitum, showed dramatic aging. And then compare that ad libitum liver profile with a wild type ad libitum. There are many genes up and down ESCC1 compared to wild type. We simply look the up genes, how big are those? And the down genes, how big are those? And these are the blue lines. And you see the down genes, they are huge. Between 100 and 1,000 kilobase, that's the maximum. So the long genes are dramatically down. And the up genes, they are a little bit shorter. So there is a tremendous dissociation between up genes, the size, and down genes, their size. And dietary restriction, which delays aging, has a curve in between. It's less down with the short genes and clearly much less down with the long genes. So there is dietary restriction reduces what we call transcription stress. Gene length dependent decline of transcription is going down with dietary restriction, and that may explain why dietary restriction delays aging. We know already for 80 years that dietary restriction delays aging, but nobody knows why. Now we see that dietary restriction lowers transcription stress, which is driven by DNA damage. That's just published in Nature Genetics. We have shown that it's DNA damage that drives this process. So we now looked, so dietary restriction actually prevents this happening. And now we looked in normal aging in humans. I'll skip the top slide. This is the hippocampus from the ESCC1 mouse. Here we just took expression profiles of hippocampi of people that had died when they were old compared to hippocampi from people that were young. There are many genes different between old and young, and simply we asked what is the size of the up genes and what is the size of the down genes with aging in the hippocampus. And this is the red curve is the up genes. They are a little bit maybe smaller than average, but the down genes, again, very big. So also in normal aging in hippocampus, the long genes go down. And not only with natural aging, we see the same in Alzheimer. So when we published this in 2016, independently, a number of other people, without knowing our results, they found not only, this is what we found, this is Nature 2016, but also in aging Drosophila photoreceptors, the same was found. In Alzheimer, other groups, particularly by coincidence in the Netherlands, they found also that particularly the long genes are the ones that go down with Alzheimer. And also this is an AI person that worked with AI, compared all the kinds of data, and he found too, with aging, long genes go down. So just yesterday in Transient Genetics, we published an opinion paper where all of the different people, there were 4 different labs with different, different approaches, and found exactly the same. Long genes go down with aging. So, and that is DNA damage driven. So the model that we have is with aging, when you are young, there is potent repair. Whenever there is a damage in a gene, there is transcription coupled repair that will eliminate it. But with aging, and you will see why, many processes will go down. And so you build up damage, and it takes longer and longer for the repair machinery to handle it. So, but particularly, of course, yeah, that's gene length dependent. If you have a long gene, there's a higher likelihood there is damage than in a short one. So you get a lower and imbalanced, particularly the imbalanced transcriptional output. That is critical. And of course, every cellular process starts with transcription. Before, you have to transcribe your genes. So you can now make the prediction, many consequences to this. And actually, we can already, from the expression profiles, predict effects on proteostasis, nutrient sensing, energy metabolism, cytoprotection, stress response, immune system, and actually, over 50% of all the changes that occur in transcription in many post-mitotic tissues, actually in all, are explained by this phenomenon, that you have long genes that go preferentially down. So, and not only in humans, even in C. elegans, in Drosophila, it's conserved throughout evolution. So, DNA damage-driven transcription stress explains most aging hallmarks in post-mitotic organs and tissues. So, the summary is aging is due to a DNA damage-driven, gene length-dependent transcriptional decline in post-mitotic tissues, neurons, liver, kidney, which cannot dilute damage or release a stalled polymerase by replication, because there is no replication. This preferentially affects long genes, leading to a lower imbalanced expression profile. Since every cellular process starts with transcription, the effects impact many pathways, and including most, if not all, hallmarks of aging. And dietary restriction redesigns metabolism. You become much more conservative. You suppress glycolysis. You upregulate the phosphate pentose phosphate shunt. You upregulate antioxidant defenses. So you boost protection systems, alleviating genome-wide DNA damage, and thereby also the transcription stress that is caused by it, and improves the gene function, the expression function, and thereby improves neural function. So the neurons were still alive, but they were struggling with a lot of damage, where the repair machine, there was too much damage that was inflicted. If you lower by calorie restriction the damage induction, then the repair machinery can still handle it. So you improve the neuromuscular junctions, and you can control your muscles better. You can speak, and you can walk. So that delays actually aging. So transcription stress affects genes stochastically, so it's random. And that will lead to lower expression of random genes. Well, if you have to build a protein complex, there are many processes in the cell that build on, you need protein complexes. So if one of the subunits has a damage and is less transcribed, then that subunit is lacking. So you get incomplete protein complexes, which affects, of course, many cellular functions, and it activates the unfolded protein response, because you need to degrade incomplete complexes, because you cannot do anything if the complex is missing some subunit. So you get proteostatic stress. So transcription stress leads to proteostatic stress. Proteostatic stress explains what was already known before, the decoupling. With aging, there is a decoupling between your expression profile at the RNA level and the protein level. They become much more apart. This phenomenon can fully explain that. You can explain the loss of stoichiometry that occurs with aging in many organs. And it explains protein aggregation. And I'll show you in a moment, because we wondered, do our mice that show a repair defect and have accelerated aging, also in the neurons, do they have actually protein aggregates? So we looked what is the effect of calorie restriction on those protein aggregates, which are, the hallmark of Alzheimer and Parkinson. So we took a protein complex that is known to aggregate in various neurological degenerative diseases. This is a mouse model for Huntington disease, and the RAN gap, it's a protein involved in nucleose cytoplasmic export, nuclear pore protein complex. We looked so this is in a mouse with a Huntington disease. You see those blebs. Those are aggregates of RAN gap that occur in Huntington patients. Of course, also in ALS patients. This is a publication from another group in 2015. Here you see those same blebs. And We found them also in frontotemporal dementia patients, and we used antibodies that in specimens of patients can visualize these blebs. RAN-GAP, Santa Cruz, polyclonal, and this is another one that also shows, and here we have SUMO that is also in the same aggregates. So we use those antibodies that in patients, frontotemporal patients, demonstrate, reveal these blebs, these aggregates. Well, now in the brain of our mice. So this is the normal wild-type mouse. There you see beautiful RAN-GAP is in a nuclear pore complex, so it decorates the nuclear envelope. But ERCC1 in another mouse that we also made, XPG, is not the same, but quite similar to ERCC1. You see dramatic blebs that occur in the brain spontaneously of these mice. So, and not only RAN-GAP is in that complex, here you see the RAN-GAP bleb, but UBC9 is in the same complex, you see in the overlay. And not only UBC9, RAN-BP2, another protein in the complex, is exactly in the same complex. So it's not only RAN-GAP, it's the whole complex that is precipitating. And not only that complex, another one, which is called the CRM1 dependent nuclear export complex, is also aggregating. Here you see RAN-GAP, this is RAN, in the same aggregate. And also CRM1 is in exactly the same aggregate. So there is no doubt that these mice develop tremendous protein aggregates. And actually dietary restriction indeed lowers them. As you can see, both in ERCC1 and XPG. Am I finished? Not yet? Can I still can I?
For your own sake, to catch your flight,
No. But I want to make it much more important. So I'll skip this. What we found that this response, that survival response, is, that boost resilience is very important for other medical applications. So surgery, if a surgeon starts to operate, clamps blood supply from the area where, because otherwise you cannot even see anything. So that area has no oxygen during the whole operation. So after the surgeon has finished, opens the arteries again, this tissue is begging for oxygen. So then you get a lot of radicals that are produced at that moment. And then you get damage, and actually that part of the body ages faster and causes for post-surgical inflammation. Very paradoxically, if you fast a few days prior to the surgery, your antioxidant defenses are already up. So at the moment then the surgeon is opening the arteries, there's a much stronger protection against the ischemia reperfusion injury. So short-term fasting prior to surgery dramatically improves the outcome. I can show you in a minute, ad libitum wild-type mice, if you clamp kidney blood supply for 37 minutes, then in 4 days, 60-70% of the mice have died. If you take the same mice and put them 1 day without food, or 2 days, or 3 days, even already 1 day of food protects dramatically from death. And 2 days and 3 days, even more. And if you look at kidney function, so you can look at urea levels in the blood. Urea is normally excreted. If your kidney is not doing well, you get sky-high levels of urea. Ad libitum animals blue, 1 day, 2 days, sky-high levels of urea. 4 days, 70% of the mice have died. Wow, if you do 1 day fasting, 2 days fasting, 3 days fasting, even stronger response. So you get a dramatic better function simply by not eating no calories before the surgery. And body weight, very interesting. Animals that had ad libitum food, they feel miserable. And then the body indicates, it's not going well. You suppress your appetite because you want to upregulate your defense mechanism. But it's too late because the damage is already inflicted. So you see these animals, they lose body weight because they have low appetite and feel miserable, and 70% died. The ones that had fasted lost body weight before. But after surgery, oh, already at 4 or 5 days, they are back to almost their normal body weight before. While these animals, almost all have died, those have all survived and do extremely much better. And if you have 2 days fasting, then the response is a little bit weaker. So the longer you fast, the stronger the survival response. Now I'll skip this. What also, very important, chemotherapy damages your DNA. And chemotherapy actually causes aging. That's why I am in the pediatric hospital in Utrecht. Children with cancer, fortunately now 85% survive because of chemo, but at the expense of accelerated aging. So 20 years later, they are 40 years older. And many of them have kidney, heart, cognition, hearing problems. So again, fasting prior to the chemo makes your body much more resilient. So we think fasting prior to chemo prevents many of the long-term effects that the chemo causes for patients. And we are now doing in the mice studies, and we want to do also studies in children. And there are more than 200 clinical trials already ongoing with adults on this principle. But interestingly, so in mice, I can show, well, maybe I should stop. But it goes at the expense of your lunch, because then less calories. Well, yeah, I'll skip this and believe it, I can only say. So we think also early Alzheimer patients, whenever, whenever patients, whenever they would be able to substantially induce calorie intake, they would upregulate their mechanism, their resilience, and they would probably improve in function and at least, delay the progression of Alzheimer. That's certainly our belief. So nutritional intervention and mimetics maybe too, they indeed have a lot of potential for medicine in the future. And hundreds of years of medical progress, but all you can tell me to do is eat less. Yes, yes. Yeah, I think there is a lot of potential there. These are our laboratory, and it's not a cook, but a look. And the mice were extremely important for this study. And please, 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 protect mouse experiments, because without mice we would have been nowhere. So many people contributed to this in the different labs that I have. People that left the laboratory, people with whom we collaborated within the Netherlands and in other countries, and also some of the funding agencies that were really supporting our research. Still, we need more, because we have still a lot of ideas to improve further. But I'm most grateful for this child. She opened our eyes for, that is translatable to patients. So I have no time probably to answer any questions. I overdid as usual. Thank you very much for your attention and for at least staying till the end.